Guys, until now we have learned about the metabolism of the compounds which are quite complex and quite bigger in size also. So, whether these are carbohydrates or lipids, these compounds are quite complex in their configuration and very heterogeneous. But today we will talk about the metabolism of the compounds which are quite simple, very small in size also. Yes, I am talking about the ketone bodies. The metabolism of ketone bodies is a very interesting subject. These molecules are interesting because their major and sole function perhaps is that they are used for providing the energy. That is their major function. And actually one of these is not even providing the energy, that is only a metabolic dead end. The metabolism of ketone bodies also have some real clinical implications. So, let us see in detail their metabolism and other aspects. So, you know that the metabolism of fatty acids produced through the I would say the phenomena or process of beta oxidation, it produces acetyl CoA, and that acetyl CoA then enters into the Krebs cycle and gives energy. But sometimes it happens that not all the acetyl CoA is oxidized. Certainly sometimes it happens that the degradation of the acetyl CoA is not as much as required. As a result of this, some new products are formed from this excessive amount of acetyl CoA, which are known as the ketone bodies. There are three important ketone bodies which we get from acetyl CoA. This is acetoacetate, beta hydroxybutyrate, and third one is the acetone. Acetone also is interesting in the sense that it acts as a metabolic dead point, dead end. I mean, it doesn't get metabolized further. It has to be excreted either through the urine, or also because of its very volatile nature, it also gets excreted through your exhaling process, through your breath. That's why the people who have excessive ketone in their blood, they give a fruity smell. So acetoacetate and beta hydroxybutyrate, once they are produced, they then are transported into the blood, in the tissues, and where then it is further oxidized to give the energy, which is the major function of these compounds. These compounds don't have as much the solubility problem as we could see when we were dealing with the long chain fatty acids. The reason is that because of they are not as much hydrophobic in nature, so they don't have to be carried by the lipoproteins or albumin. They are mainly produced in the liver, and when the oxidative capacity of liver exceeds, what will happen, that these ketone bodies are produced. So, as I mentioned that they are used in different tissues, in skeletal muscles, and sometimes brain also use these compounds specifically, especially when the blood level of these bodies is higher in the blood, then the brain also use them as a source of energy. Also, I have already told you that when you are facing starvation or you are undergoing prolonged fasting, then the body moves from the glucose based metabolism towards the lipid based metabolism, and here the ketone bodies serve really very important purpose, because they are mainly, their major function is to provide the energy. Now let's we talk about the synthesis of the ketone bodies. First of all, as you must keep in mind what are the compounds which are basic ketone bodies. As I have already told you, these are three compounds: beta hydroxybutyrate, acetone. These are main compounds which are acting as the ketone bodies. So, if we see here, first of all, this is the acetyl CoA. Two acetyl CoA compounds, they combine with each other to make the acetyl CoA, and this acetyl CoA, under the action of synthetase enzyme, this is HMG CoA synthetase, that is hydroxymethyl CoA synthetase, it gives rise to HMG CoA. This HMG CoA then undergoes further reactions to give us the first ketone body, that is acetoacetate. The enzyme used here is the lyase enzyme. Acetoacetate then further undergo modification to give rise to two more ketone bodies. One is the acetone, which is the metabolic dead end, and the 3-hydroxybutyrate. So, in this way, you learn that how the three major ketone bodies are produced. Acetoacetate first is formed, and then that acetoacetate gives rise to the formation of acetone and 3-hydroxybutyrate. And also you can see, because it's a biosynthetic process, it requires energy, and here NADH is used. Also keep in mind that the rate limiting step here is the when acetyl CoA is changed into HMG CoA, and this enzyme, the HMG CoA synthetase, it actually is the major regulatory enzyme as far as the formation of the ketone bodies is concerned. Once these ketone bodies are formed, they are then released and they go into different tissues, and they are then used for the production of the energy. So, this is the repetition of the same thing which as I have already told you, that acetoacetate give rise to acetone and hydroxybutyrate. Yes, one thing here is important you should know, that the equilibrium will tell that to which direction the reaction will move. That is the ratio of NAD by NADH2. For example, if you have excessive NADH, definitely the excessive NADH means that it can be utilized for the formation of the ketone bodies. But if the the ratio between NAD and NADH is in a way that NAD is more than NADH, it will definitely then lead to lesser formation of the ketone bodies. It will inhibit the ketone body formation. So, as I have told you, the ketone bodies are formed in the liver, and you know the liver is such an important organ. We are seeing it again and again, that majority of the biochemical pathways depend upon the functioning of the liver. So, the ketone bodies, when formed, they move towards your tissues where it is required. For instance, they come into the skeletal muscles, and here 3-hydroxybutyrate is converted into acetoacetate, and the acetoacetate, and of course you get the NADH, which provides you the energy because it goes into TCA and gives you the ATP. The acetoacetate then get a CoA from the succinyl CoA from the Krebs cycle, because it's happening in the mitochondria. So, you get these compounds or intermediates of Krebs cycle very easily during the catalysis or the degradation of the ketone bodies, and you get from acetoacetate to acetoacetyl CoA, and then acetoacetyl CoA is changed into its CoA moiety is released, and you get 2 acetyl compounds, and you know that acetyl CoA is then further degraded in the process of Krebs cycle, and you get more and more energy.